Good evening, everyone. Tonight we will discuss the motion. Animal testing is necessary for scientific progress. We have one debater representing the affirmative team on my left, and one debater for the negative team on my right. Here is how our debate will proceed. First, the affirmative team will present their opening statement, followed by the negative team's opening statement. After that, we will have the first cross-examination, where the negative team will question the affirmative. Next, both teams will present their rebuttals. We will then have the second cross-examination, with the affirmative team questioning the negative. I will provide a brief time check, and then each team will deliver their closing statements. Finally, I will thank our debaters and conclude the event. Let us begin.
Thank you, Madam Moderator. Esteemed judges, ladies and gentlemen, I stand before you tonight to unequivocally affirm the motion. Animal testing is necessary for scientific progress. We are not here to debate the ethics of suffering, but the undeniable reality that the most profound leaps in medicine and our understanding of biology have, and continue to, rely on the use of animal models. Animal testing provides an invaluable bridge between theoretical knowledge and practical application, allowing scientists to study complex biological systems in a living organism that cannot be replicated by petri dishes or computer simulations alone. It is through these studies that we gain critical insights into disease mechanisms, test the safety and efficacy of new drugs, and develop life-saving vaccines and surgical techniques. From the eradication of smallpox to the development of insulin, chemotherapy, and organ transplantation, countless human lives have been saved and improved because of the knowledge gained through animal research. While we all strive for a future with fewer animal tests, the scientific reality is that, today, these studies remain an indispensable tool for advancing human health and scientific understanding. We will demonstrate how animal testing is not just a historical footnote, but a current necessity for continued progress.
Thank you, Madam Moderator. Esteemed judges, ladies and gentlemen, the affirmative team has presented a compelling historical account, highlighting past achievements linked to animal testing. However, the motion before us today is not about the historical role of animal research, but whether animal testing is necessary for scientific progress now and into the future. We, the negative team, firmly contend that it is not. While acknowledging the past, we must critically examine the scientific validity and ethical implications of these methods in our current age of rapid technological advancement. Animal models, despite their historical use, frequently fail to accurately replicate human physiology and disease, leading to misleading results, countless failed drugs, and ultimately, a significant impediment to genuine, human-relevant progress. To continue relying on methods that often provide unreliable data, divert crucial resources, and raise profound ethical questions is not true progress, it is stagnation. True scientific progress lies in embracing innovative, human-specific research methods, advanced in vitro models, organ-on-a-chip technologies, sophisticated computational simulations, and human-based microdosing, which are proving to be more predictive, more efficient, and undeniably more ethical. We will demonstrate how these modern approaches are not only viable alternatives, but are actively rendering animal testing obsolete, proving it is a relic of the past, not a necessity for our scientific future.
Esteemed judges, ladies and gentlemen, the negative team attempts to dismiss the historical successes of animal testing as mere relics, irrelevant to our present and future. But this is a grave misrepresentation of scientific reality. While we embrace the promise of advanced alternatives, the critical word is promise, not current full replacement. The negative team asserts that animal models frequently fail to replicate human physiology. Yet they conveniently overlook that these models, despite their limitations, are the most sophisticated whole-system biological models we currently possess. They are not perfect, no model is, but they are indispensable for understanding complex systemic interactions, long-term effects, and the pharmacokinetics and toxicology of new compounds within a living, integrated organism, aspects that no petri dish or algorithm can yet fully replicate. Failed drugs are a reality in scientific discovery, but it is often animal testing that helps us identify these failures before they reach human trials, saving countless lives and preventing harm. To suggest this is a diversion of resources is to ignore the fundamental role of preclinical research in ensuring safety and efficacy. Furthermore, the very innovative, human-specific research methods the negative team champions be it organ-on-a-chip technology, advanced in vitro models, or sophisticated computational simulations, are often developed, refined, and even validated against data derived from animal studies. They are not independent solutions, but powerful complementary tools that work alongside, not entirely replace, the necessity for whole organism studies, particularly when moving from basic research to clinical application. Until these alternatives can fully replicate the entirety of human physiology, disease progression, and therapeutic response at a systemic level, animal testing remains a crucial, albeit continually refined, necessity for current and future scientific progress.
Esteemed judges, ladies and gentlemen, the affirmative team attempts to diminish our arguments by clinging to the idea that animal testing, despite its admitted limitations, remains the most sophisticated whole system model. This line of reasoning fundamentally misunderstands the very nature of scientific progress. To suggest that an approach which frequently fails to predict human outcomes is somehow sophisticated, simply because it is a whole organism, is to mistake complexity for accuracy. The very failures they concede, the failed drugs they mention, are not always caught efficiently by animal tests. Often, they only become apparent during costly and risky human trials after animal models have misled researchers. This is not saving lives efficiently. It is a costly and often tragic detour. We are not talking about models that are not perfect. We are talking about models that are frequently not relevant to human physiology and disease. The affirmative team's insistence on a whole system approach at all costs, even when that system is fundamentally different from a human one, is precisely why we must move on. Furthermore, to claim that our innovative, human-specific research methods are merely a promise or are only useful because they are validated against animal studies is a profound mischaracterization. These methods are not just promises, they are current realities already delivering superior, human-relevant data in countless research areas. And the validation they speak of is often an initial benchmarking phase, a stepping stone, not a perpetual dependency. The ultimate validation for human medicine must come from human biology itself, not from species that react differently to diseases and treatments. To argue that we must wait for these alternatives to fully replicate the entirety of human physiology before discontinuing animal tests is to set an impossibly high and unnecessary bar. Our goal is not to perfectly replicate an entire human in a lab, but to develop models that are more predictive and more relevant for specific human diseases and drug responses than animal models currently are. And in many cases, we are already there. Animal testing is not a necessity for future scientific progress. It is an impediment that is actively being surpassed.
Esteemed judges, ladies and gentlemen, the negative team proposes that animal testing is a impediment, and that alternatives are already delivering superior, human-relevant data. While we acknowledge the incredible advancements in alternative methods, this statement paints an overly optimistic and frankly inaccurate picture of their current capabilities across the entire spectrum of scientific inquiry. The negative team suggests we needn't replicate an entire human, and we agree, that's not the goal. However, understanding how a drug is metabolized and eliminated by a liver, how it interacts with the immune system, affects the central nervous system, and impacts other organs simultaneously and over time. That is the essence of whole system biology, and it is precisely where current alternatives still fall short. Consider vaccine development, a monumental achievement in public health. The efficacy and safety of new vaccines, such as those for COVID-19, fundamentally rely on testing within a living organism to understand the complex immune response, the protection conferred against infection, and potential systemic side effects. Organ-on-a-chip technologies are incredible for studying specific organ functions or drug toxicity in isolation, but they cannot yet replicate the intricate interplay between a spleen, lymph nodes, bone marrow, and the circulating immune cells in response to a pathogen. Similarly, for complex neurological conditions like Alzheimer's or Parkinson's disease, understanding the progression of pathology, the behavioral changes, and the long-term efficacy of potential therapies often requires an integrated living system. To dismiss animal models as an impediment is to ignore the ongoing critical research areas where they are still the only available tool for obtaining this holistic, systemic data. Animal testing is not a historical footnote, it is a necessary, albeit constantly refined, bridge for current scientific progress, allowing us to ask and answer questions that no current alternative can truly address.
Esteemed judges, ladies and gentlemen, the affirmative team continues to frame the necessity of animal testing around a perceived lack in alternative methods, claiming they are overly optimistic or fall short in providing holistic, systemic data. This argument, however, fundamentally misses the critical distinction. The problem is not a lack of a whole system, but rather the profound irrelevance of the wrong whole system. Why should we prioritize holistic data from an animal whose physiology, immune response, and disease progression are fundamentally different from our own, when that data so often fails to translate to humans? Consider the very examples raised: vaccine development and neurological conditions. While animal models have historically been used in vaccinology, modern vaccine research, especially exemplified by the rapid development of COVID-19 vaccines, relied heavily on human genomic sequencing, advanced in vitro human cell culture models, and sophisticated computational analysis to understand human immune responses, not solely animal models, which often exhibit species-specific immune reactions. Animal testing in this area frequently leads to false positives and negatives, making it a less, not more, reliable predictor for human efficacy and safety. Similarly, for complex neurological conditions like Alzheimer's and Parkinson's, decades of research using animal models have yielded countless failures in human clinical trials. These animal models simply do not accurately replicate the human disease pathology or response to treatment. In contrast, human brain organoids derived from induced pluripotent stem cells, human postmortem brain studies, and advanced human imaging techniques are now providing unprecedented insights into human-specific disease mechanisms, insights that are far more relevant and predictive. The notion that animal models are the only available tool for holistic, systemic data in these critical areas is demonstrably false and overlooks the significant, human-relevant advancements that are actively surpassing their utility. We are not waiting for a single alternative to perfectly mimic an entire human. We are strategically employing a suite of human-specific, sophisticated technologies that collectively provide superior, more predictive data for human health than animal testing ever could.
Thank you both for your insightful arguments. We have reached the final stage of our debate. We will now proceed with the closing statements. Each team will have 2 minutes to deliver their final thoughts. Affirmative team, please prepare your concluding remarks.
Thank you, Madam Moderator. Esteemed judges, ladies and gentlemen. Throughout this debate, we, the affirmative team, have consistently demonstrated why animal testing remains a necessary cornerstone for scientific progress. We are not advocating for the suffering of animals, but for the undeniable scientific reality that, for many complex questions, animal models are currently the only available tools to bridge the gap between theoretical understanding and practical, life-saving applications for human health. We have shown how critical breakthroughs, from vaccine development to the treatment of chronic diseases, have fundamentally relied on insights gained from living, integrated systems that cannot yet be replicated by alternatives. While we embrace the promise and potential of these evolving technologies, it is a scientific fallacy to suggest they are, today, full replacements for the nuanced, systemic data derived from animal studies. Alternatives are powerful complements, often refined and validated through the very animal research they seek to eventually reduce. Until these sophisticated alternatives can fully replicate the intricate interplay of organs, immune systems, and long-term physiological responses within a whole organism, especially for chronic diseases and the systemic safety and efficacy of novel therapies, animal testing remains not just a historical success, but a current and future necessity. To abandon it prematurely would be to hinder, not advance, the very progress we all seek for humanity. We stand firm. Animal testing is necessary for scientific progress.
Thank you, Madam Moderator. Esteemed judges, ladies and gentlemen, throughout this debate, the negative team has rigorously challenged the notion that animal testing is necessary for scientific progress. We have consistently argued that while animal models hold a historical place, their continued reliance in the face of scientific advancement is no longer a necessity, but an impediment. We have demonstrated how species differences frequently render animal data unreliable and poorly predictive for human outcomes, leading to countless failures in human clinical trials and a tragic diversion of resources and scientific effort. True progress, we contend, lies in embracing the sophisticated human-specific research methods that are not merely promises for the future, but current realities. Technologies such as human organoids, advanced in vitro models, computational simulations, and human genomic sequencing are delivering far more relevant, accurate, and ethical data for understanding human disease and developing human therapies. To cling to outdated methodologies when superior human-relevant alternatives are actively surpassing their utility is to stifle innovation, not to foster it. We must move beyond the limitations and ethical concerns of animal testing and fully invest in the pathways that genuinely advance human health. Animal testing is not necessary for scientific progress. It is a relic being superseded by the ingenuity of modern science. We urge you to reject the motion.
Thank you to both the affirmative and negative teams for your compelling arguments and insightful presentations throughout this debate. Your thoughtful exploration of the motion, animal testing is necessary for scientific progress, has been truly enlightening. We appreciate your participation and the valuable perspectives you have shared with us tonight.